So in this lecture on organization and physiology of the autonomic nervous system, I want to first give you an introductory aspect of the nervous system. We divide the nervous system anatomically into two. There's what we call the central nervous system, and there's what we call the peripheral nervous system. When we talk of the central nervous system, we are primarily referring to the brain and the spinal cord. So this is the brain located within the cranial cavity under the spinal cord, located within the vertebral column. Those two constitute the central nervous system. When you talk of peripheral nervous system, we are referring to nerves. What is a nerve? A nerve is a group of neuronal processes outside the brain and the spinal cord. A group of neuronal processes outside the brain or spinal cord. That's a nerve. Remember, these neuronal processes could be axonal processes. They could be dendritic processes, or they could be both. Whether they are axonal or dendritic or both, as long as a group of processes, we call that a nerve. We name the nerves based on where they come from. The ones that come from the brain are known as cranial nerves. And we have 12 pairs of them. The first pair is the olfactory nerve. That's the cranial nerve number 1. Number 2 is optic nerve. Number 3 is oculomotor nerve. Number 4 is trochlear nerve. Number 5 is called trigeminal. Number 6 is abducens. 7 facial. 8 vestibulocochlear. 9 glossopharyngeal. 10 vagus. 11 accessory. And 12 hypoglossal nerve. In this lecture on the autonomic nervous system, 4 of those nerves will be very important. So we'll not talk about all of them, only 4 of them. So those are cranial nerves, 12 pairs. They arise from the brain, or at least they're attached to the brain. The nerves which come from the spinal cord are called spinal nerves. While the cranial nerves are 12 pairs, the spinal nerves are 31 pairs. We name them as well, but the naming system is simpler than that of the cranial nerves, so it's easy to remember them. We name them according to where they come from. The ones which come from the cervical segment of the spinal cord, we use the letter C to refer to cervical. There are 8 pairs. So we talk of C1, C2, C3, all the way to C8. The ones which arise from the thoracic segment are 12 pairs. We use the letter T to refer to thoracic. So we name them T1, T2, all the way to T12. The ones arising from lumbar segments are 5 pairs. We use letter L to refer to lumbar, so we talk of L1, L2 up to L5. The sacral, we use S, so we name them S1 up to S5. The coccygeal segment has only 1 pair of spinal nerve. So if you add those ones in total, we have 31 pairs of spinal nerves. Again, not all those 31 pairs of spinal nerves will have autonomic functions. We will see which ones actually do have autonomic functions. But before we go far, perhaps you need to understand, so what's the role of the nervous system? The nervous system primarily coordinates and integrates body functions. This coordination and integration of body functions tend to occur in a particular operational model that I want to describe for you. So let's start it this way. We have what we call sensory receptors. The role of the sensory receptors is to detect stimuli in the environment, the changes which are taking place in the environment. It could be temperature changes, it could be pain, it could be vibration, it could be light, it could be sound. Any stimuli which is in the environment may be detected by sensory receptors. Fundamentally, what sensory receptors do is to convert the environmental stimuli in whichever form that stimulus comes in. The sensory receptors convert them to action potentials. So fundamental that if something can convert environmental stimuli into action potentials or nerve impulses, it definitely qualifies to be called a sensory receptor. And if something cannot transduce environmental energy into nerve energy, then it doesn't qualify to be called a sensory receptor. Very fundamental. Now, this becomes fundamental because the only language that the central nervous system, or basically the nervous system, understands is action potentials, or basically nerve impulses. And remember, they are bioelectric impulses. They move along the axons of neurons. So whether it is light, whether it is sound, whether it is pain, whether it's touch, whether it's temperature, the information that goes to the brain is nerve impulses. So sensory receptors must be able to convert the whatever form of physical energy into nerve impulses. Once they have converted the physical energy to nerve impulses, we have a group of neurons that carry those information, those impulses, to the central nervous system. The name given to the group of neurons that carry the impulses from the receptors to the central nervous system is sensory neurons. It's not just one neuron, it's a group of several neurons. So we just call them sensory neurons, or basically ascending tracts because they go up. You can call them ascending pathways. Why do they go to the central nervous system? Among many reasons why these impulses would travel to the central nervous system, I'll give you three fundamental ones. One of them is that the impulses need to be interpreted. So that if it is impulse coming from the retina, for example, then that will be interpreted as light. If it's an impulse coming from the ear, it may be interpreted as sound. If it's an impulse coming from pain receptors, it should be interpreted as pain. Remember, whatever went into the brain is electrical impulse. All of them are electrical impulses. But now the brain needs to interpret that electric impulse to mean pain or light or touch, depending on where that impulse has originated from and where that impulse is going to the brain. So the brain interprets the sensation, and this is what will give you perception, so that you're able to perceive that particular sensation. Again, so fundamental that if that region of the brain is injured, then you may even be burnt with fire. and you'll just be smiling. You won't feel any pain. The brain must interpret the sensation. Other than interpretation, the central nervous system also keeps memory of the sensation that has entered it. So for example, if a child was playing with fire, they don't have to do that several times. They'll remember that this thing is not good because of what it has given them. There's memory that is established in the central nervous system. The third thing I want to say is that the central nervous system provides a response. This response is with regard to the type of sensation that has gone in. And depending on how the brain has interpreted it, there'll be a reaction that the central nervous system provides. When the central nervous system provides a reaction, that reaction must go back to what we call effector organs. Effector organs are organs that execute the responses which the central nervous system has initiated. These effector organs can either be muscle or they can be glands. And remember, if they are muscles, then maybe they could be smooth muscles, they could be skeletal muscles, and they could also be cardiac muscles. So the muscles are different types: smooth muscles, cardiac muscles, and skeletal muscles. All those are muscles. Apart from muscles, they could be glands. Remember, muscles respond by contracting. Then glands respond by providing a secretion. Those are the kind of responses that the central nervous system initiates to go to the effectors. The group of neurons that come from the central nervous system to the effectors are the motor neurons. In our earlier classes, we may have called them efferent neurons, and that's still okay. Because they come from the central nervous system going to the effectors, we commonly call them the descending pathways, as opposed to these ones which are called the ascending pathways. Again, it may not necessarily be just one neuron, but it could be a relay of multiple neurons. And that concept will be discussed more when you look at the nervous system in general. So for now, I want you to understand that operational model of the central nervous system, or rather of the nervous system, that help the nervous system to coordinate and regulate body functions. Remember, some of these sensory neurons would actually be part of the peripheral nervous system, but some of them are within the central nervous system either way. Similarly, some of these motor neurons are in the central nervous system, but there are those that are also within the peripheral nervous system. They're already outside the brain and spinal cord. So they could be carried within the cranial nerves, these sensory and motor nerves. They could be within cranial nerves, or they could be within spinal nerves. Definitely the effectors are not within the CNS, and the sensory receptors are also not necessarily within the CNS. I want to remind you something as we talk about the signals being transmitted within the central nervous system. And we are talking about impulses moving from the sensory receptors to go to sensory neurons. And I've told you that it's not necessarily one neuron, but a relay of multiple neurons, which means that impulses travel from one neuron to another. When impulses travel from one neuron to another, there'll be need for impulses to be transmitted between junctions of neurons. And in our earlier class, we talked about junctional transmission in neurons. We talked about two types of junctional transmissions: electrical junctions which uses gap junctions, and those ones are straightforward. Depolarization in one cell spread to another adjacent cell because of movement of charged particles within the depolarized cell into the one that's not yet depolarized, but now it's going to be depolarized. And so impulses move quickly. Those are electrical junctions, via gap junctions. We have also the chemical junctions. The chemical junctions use synapses. Use synaptic clefts. There's some synaptic clefts. They use neurotransmitters to transport the impulses from one to another. Now, let's review how the impulses actually move from one neuron to another across a chemical synapse. We talked about an action potential arriving at the end of an axon. The end of an axon is what we called sometime back the axonal terminal. When that action potential arrives at the axonal terminal, remember it's a depolarization sequence. That depolarization sequence causes the opening of voltage-gated calcium channels, which are at the axonal terminal. When we open the voltage-gated calcium channels, and we know that calcium is more outside than in the cytoplasm. Well, even if calcium inside the cell may be more, usually it's enclosed within the SER. So the free calcium in the cytoplasm is actually little compared to the calcium that is in the extracellular space. For that reason, if you open the voltage-gated calcium channels, we'll have calcium influx into the axonal terminal. The entry of calcium into the axonal terminal causes the synaptic vesicles to move from where they are to the presynaptic membrane. Remember, the synaptic vesicles contain the neurotransmitter chemicals. So if the synaptic vesicles move from where they are to the presynaptic membrane, they attach to some proteins there. That process is called docking. And so those proteins are called the docking proteins. Basically, the point here is that the synaptic vesicle go and attach to the presynaptic membrane. The next step would be that the synaptic vesicle releases its chemical into the synaptic cleft. That process of the synaptic vesicle emptying its neurotransmitter chemical, the chemical it's containing into the synaptic cleft, is exocytosis. So the process of releasing the neurotransmitter is through exocytosis. Once the neurotransmitter chemical is within the synaptic cleft, there are a number of things that can happen to the neurotransmitter chemical. And in our earlier lecture, we talked about some 3 or 4 things that can happen to the neurotransmitter chemical. One of them was that the neurotransmitter chemical can actually be degraded by an enzyme. And so that becomes the end of that story. If the neurotransmitter chemical undergoes enzymatic degradation, it ends there. The way acetylcholine can actually be broken down by acetylcholinesterase enzyme. The other thing is that the neurotransmitter chemical may be taken away from the synaptic cleft by other cells which are surrounding. if you are in the central nervous system, those other cells could be astrocytes, for example. And that process of neurotransmitter chemical moving from the synaptic cleft into another surrounding cell is what we call the uptake process. So it may be uptaken by glial cells. Again, you realize that does not contribute to signal transmission in this case. The third option is that the neurotransmitter chemical may actually go back to the neuron that released it. So if the neurotransmitter chemical is absorbed back by the cell that released it, we gave that a name as well, and we call that the reuptake process. Again, the reuptake process does not contribute to signal transmission. So the only event that contributes to signal transmission is if this neurotransmitter chemical binds to its receptor on the postsynaptic cell. And so yes, the neurotransmitter that has been released into the cleft would bind to its receptors on the postsynaptic cell. When the neurotransmitter binds to its receptor on the postsynaptic cell, what does it do to the receptor? These receptors are ligand-gated sodium channels. I hope you remember the different types of channels: voltage-gated channels, ligand-gated channels, mechanically gated channels, and leaky channels. Leaky channels are always open, so they just allow diffusion of substances freely, anytime. Gated channels are usually closed, but they can be triggered to open. Based on the type of trigger, we give them different names. So the ones which are triggered by voltage disturbance are called voltage-gated channels. The ones which are triggered by binding of a chemical messenger are known as ligand-gated channels. So here we have a chemical messenger named neurotransmitter, which binds onto its receptor on the postsynaptic cell. The binding of the neurotransmitter to the postsynaptic cell make this receptor to open. Therefore, that receptor is a ligand-gated channel. So let's see here. They are ligand-gated sodium channels, which means that they allow sodium to pass through them once activated to open. So what would happen is that if you open the ligand-gated sodium channels, then sodium ions will enter into the cell. In this case, sodium ions enter into the postsynaptic cell. The entry of sodium into the postsynaptic neuron will cause depolarization in the postsynaptic neuron. And if you have depolarization in the postsynaptic neuron, it may reach the threshold that will initiate an action potential. So therefore, we see that an action potential came in the presynaptic neuron. A lot of molecular events have taken place. And now we see another action potential being initiated in the postsynaptic neuron. That is how junctional transmission takes place in neurons when you look at the chemical synapses. That story of chemical synapses is very important in this particular lecture because we'll be talking about neurotransmitters and we'll be talking about receptors. We'll be talking about autonomic neurotransmitters and we'll be talking about autonomic receptors. And so it's important that we remember that story, and that's why I've taken my time to remind you about the same. Okay, nervous system can also be divided in a functional way. Functionally, we divide the nervous system again into two. Remember, the first one was an atomic division. And we talked about the central and peripheral nervous system. In this division, which we are calling functional division, we recognize two wings of the nervous system. The sensory division, concerned with passing impulses to the central nervous system, and motor division, concerned with execution of responses from the central nervous system. Based on that, we can classify the sensory division according to the types of sensations which are there. On that note, we have what we call spatial sensations and general sensations. The main difference between spatial sensations and general sensations is this. Spatial sensations are housed within the head region exclusively. So think of sensations which are exclusively within the head region. You can't find them anywhere else. And these spatial sensations are only mediated by cranial nerves. So the impulses from those receptors are only relayed by cranial nerves, not spinal nerves. Those are spatial sensations. There are 5. So the sense of sight from the retina, the sense of smell from the nose, the sense of taste from the mouth, the sense of hearing or auditory function from the ear, and the sense of equilibrium from the ear, which we call vestibular function. Those ones are spatial sensations. How about the general sensations? So they are not limited to the head region, which means they are found even the rest of the body. Even if they are found in the head region, they are also found elsewhere else. And 2, even if they may be traveling by cranial nerves, they also travel by spinal nerves. Those are general sensations. There are many. Sense of pain, heat, cold, touch, pressure, vibration, and proprioception. Proprioception is a sense of position, the sense of spatial orientation or orientation in space. Even if you close your eyes when you're seated, you are still aware that you are seated. You don't have to open your eyes to confirm that you are seated. And you can stand with your eyes closed and you know that you're actually standing. You are aware of your orientation in space. That is proprioception. So that is a sensory division, not the subject matter of today. How about the motor division? The motor division is divided into 2 based on how the central nervous system control the effectors. If the central nervous system is controlling the effectors from a voluntary perspective, which means it is within the consciousness of the individual, then we call it somatic nervous system. So that the somatic division of the nervous system is the division of the nervous system that is within the voluntary control, within our conscious control. That particular division of the nervous system targets the skeletal musculature. If you remember, skeletal muscles are voluntary. The other division of the efferent wing is what we call the autonomic nervous system, which is the subject matter of today's talk. The autonomic division of the nervous system is concerned with involuntary functions. Functions which are subconscious, functions that you do not have conscious control about, but they still happen. The autonomic division targets 3 types of effectors. You have smooth muscles, you have cardiac muscles, and you have glands. So this is the subject matter of today's talk, the autonomic nervous system. Let's say something about it. So if we are to define what is autonomic nervous system, therefore, we'll say is the division of the nervous system that is concerned with some conscious functions. This division of the nervous system have neurons which terminate within some 3 types of effectors: smooth muscles, cardiac muscles, and exocrine glands. So remember, cardiac muscles will respond by contraction and relaxation. Smooth muscles will also respond by contraction and relaxation. Exocrine glands respond by providing secretion. Think for a moment where we already know where the cardiac muscles are. They're found on the heart. Think for a moment where the smooth muscles are, and think for a moment where the exocrine glands are. So you'll realize that the smooth muscles are found within the walls of internal hollow organs, like the blood vessels will have smooth muscles. The digestive tract has smooth muscles. Respiratory tree has smooth muscles on the wall. Urogenital system, the urogenital tracts have smooth muscles. Exocrine glands are those glands which provide body fluid secretion into ductal system. So things like salivary glands, sweat glands, lacrimal gland, pancreatic acid cilia, and many other exocrine glands qualify in this category. Right. Why do we really need the autonomic nervous system? The autonomic nervous system is responsible for homeostasis of body functions. It controls body functions and keep the parameters within physiological ranges. For example, you don't have control of your body temperature so much. The autonomic nervous system does it for you. It's able to keep your body temperature within some physiological range. You also don't have so much control over your blood glucose levels. The autonomic nervous system does it for you. Of course, there's that part that you have to play, like eating or fasting. But the exact level, the autonomic nervous system will be doing it for you. So basically, that's what I mean by homeostasis of body functions. It keeps some body functions within physiological parameters. We realize that you don't really need autonomic nervous system to live. Let me say that in a different way. It may not necessarily be vital for life. However, for practical purposes, you need it so that you can adapt to environmental changes. So if it was possible for us to put you in a particular environment where the parameters are constant, you may not need the autonomic nervous system. But we know that's not the reality. The reality is we live in an environment where there are changes anytime. Even right now, when you're listening to me, Perhaps when you started, the sun was somewhere and now the sun has changed. And so there's some change in the environmental temperature. That's an environmental stressor. If we couldn't be able to have the autonomic nervous system, if you didn't have autonomic nervous system, it means that any change in the environmental temperature will just be translated directly onto your body. For example, if you are to subject you to lower environmental temperature, your body temperature will just go low. So imagine when that time when the environmental temperature is 10 degrees, it means your body will threaten to reach that level if you don't have autonomic system. And when the sun is scorching, maybe it's 40 degrees, 45 degrees, perhaps that's what your body temperature will threaten to arrive at as well. And so you realize in that case, you'll actually not survive because you'll not be able to adapt to the environmental stresses. And so the cause of death would be because you are not able to adapt to the extreme of the environmental changes. That therefore makes it a vital system in us in the understanding that we cannot, it is not possible to live in an environment where there are no stresses. Okay. Now, let's talk about chemical transmissions at autonomic synaptic junctions. Now I'm bringing this again so that we remember that yes, we talked about chemical synapses that take place within neurons, but which are these synapses when you're talking about the autonomic nervous system? Which are the synaptic junctions? The synaptic junctions, which are in the autonomic nervous system, are synaptic junctions between what we call preganglionic neuron and postganglionic neuron. And we are going to define what is preganglionic and what is postganglionic shortly. The place where those synapses is known as autonomic ganglia. We also have synaptic junctions between that postganglionic neuron and the effector organ. We agreed it could be smooth muscle, cardiac muscle, or exocrine gland. The synaptic junctions between the postsynaptic neuron and the organs are also the target synaptic sites that we are going to talk about. The main neurotransmitters that are in the autonomic nervous system are these two: acetylcholine and norepinephrine, which is also known as noradrenaline. Well, there are other neurotransmitters as well in some sites, but the principal ones are these two, and those are the ones we'll be talking about more in this particular class. Okay, having given you all that, so let me tell you how the autonomic nervous system is divided. The autonomic nervous system has two divisions. We have what we call the sympathetic division, and we have what we call the parasympathetic division. These two divisions control basically the subconscious functions. In terms of how they control the effector organs, there's two concepts here that I want us to highlight. There are cases where the two divisions both supply a target organ. We call that dual innervation. Dual innervation simply means that that organ receives both sympathetic as well as parasympathetic innervation. But there's also what we call single innervation. Single innervation is where an organ is controlled only by one division. In most cases, the sympathetic division is the one controlling it. So you may have single innervation and you may have dual innervation. In the case of dual innervation, there's something I want to clarify. You may have dual innervation with antagonistic effects, which means what? That the effect of sympathetic is in the opposite direction as that of the parasympathetic. If one lowers, the other one raises. If one makes it faster, the other one makes it slower. That is dual innervation with antagonistic effects. So sympathetic and parasympathetic may both innervate an organ, so that is dual innervation. But they have opposite effects. So we call that dual innervation with antagonistic effects. You may also have a scenario where both divisions supply a particular organ and they have different effects. So we call that dual innervation with different effects. Different here does not necessarily mean that they are in the same direction or opposite direction. They're just unique, different things. And I'll be giving examples of dual innervation with different effects. Third category is that you may have dual innervation with synergistic effects. That means that the physiological effects that we see are synergistic. They help one another. Again, I'll cite for you an example where we have dual innervation with synergistic effects. So I want you to remember those, depending on how you count them, 4 types of innervations. One is single innervation, which means it's receiving either sympathetic or parasympathetic, but I've told you in most cases, just sympathetic. There's no parasympathetic nerve supply to that particular organ. That is called single innervation. The second one is the dual innervation with antagonistic effects. And that's the majority. Most organs are under dual innervation with antagonistic effects. The third one is dual innervation with different effects. A few fall under that category, and I'll give you an example. And lastly, dual innervation with synergistic effects. Again, I'll cite an example with that regard. Okay, there's another division of the enteric nervous system, of the autonomic nervous system, that to be worth mentioning, although we'll not talk about it much because it is subject matter of the GIT physiology. But just to say something about it, there's a division of the autonomic nervous system that we call the enteric nervous system. This is the division of the autonomic nervous system that control the functions of the digestive system. It is somewhat independent, and that's why we prefer to give it its own special mention. So in this lecture, we'll mention it a bit, but much of it we'll talk about if you look at the anatomy and physiology of the digestive system. So these images show us on your right-hand side the parasympathetic innervation to different organs, and on your left-hand side, sympathetic division or innervation to different organs. What we are able to capture at a glance is that whether sympathetic or parasympathetic, they arise from the central nervous system. At least we can pick that one from here, we can pick that from here. We see the brain and the spinal cord and nerves coming out of it. Or here, brain and spinal cord, and we see nerves come out of it. But one big difference we can pick from this glance is this. Neurons which are parasympathetic, your right-hand side, either come from the brain stem, which means they are cranial nerves, or they come from the sacral segments of the spinal cord. So they are either cranial nerves or sacral spinal nerves. To your left-hand side, what do you see? The neurons which are sympathetic in function either arise from thoracic segments of the spinal cord or the upper lumbar segments of the spinal cord. So either thoracic spinal nerves or lumbar spinal nerves, and it's not all the lumbar spinal nerves, just the upper lumbar. All in all, the type of effectors we see in this image seems to be roughly similar to the type of effectors we're just seeing on this other image. Of course, we'll highlight on what are some of the unique differences. So having given you that, that was introduction actually. Having given you that, now I can give you the learning outcomes, then maybe allow you to ask a few questions before we proceed. So the major objective of this lecture is first to describe the anatomic organization of both sympathetic and parasympathetic divisions of the autonomic nervous system. Then we look at the neurotransmitters. We've already mentioned acetylcholine and norepinephrine. But we also want to look at the receptors of those neurotransmitters. Which receptors of acetylcholine are we talking about? Which receptors of noradrenaline are we talking about? Because that becomes vital in the understanding of pharmacology of the autonomic nervous system. Then we'll talk about the physiological effects that the two divisions have on different body organs. We will then perhaps talk about the components, features, and role of the enteric nervous system, although that will be just a single slide. And we'll not do that one though, but I'll give you a chance to talk about the differences between autonomic and somatic divisions of the nervous system. So that is our objective of this lecture. We are now going to talk about the general organization of both divisions of the autonomic nervous system. So this organization is general, whether it is sympathetic or parasympathetic division. Both sympathetic and parasympathetic divisions have a relay of two peripheral processes. This is what I mean. If we consider this to be the brain and this other one to be the spinal cord, and we consider this to be one of the target organs, in this case, the heart, irrespective of how it looks like, the heart is one of those organs that enjoy dual innervation. So we can use it to see how both sympathetic and parasympathetic are organized. We are saying from the central nervous system, to the effector organ, we have a relay of two peripheral processes. What does that mean? It means that not just one single neuron that will come from the central nervous system to the effector, but rather there'll be a neuron that is coming from the central nervous system that will synapse with another neuron that will then reach the effector. The name given to the neuron that come from the central nervous system is preganglionic neuron. The name given to the neuron that reaches the effector is postganglionic neuron. And that does not matter whether the neuron was coming from the brainstem or it was coming from the spinal cord. The same concept we will have. The preganglionic neuron is from the central nervous system and it will synapse with another one. Then postganglionic neuron will be the one reaching the effector organ. Why do we use those terminologies ganglionic? This is the answer. The term ganglion refers to a collection of neuronal cell bodies outside the central nervous system. So here we have a collection of cell bodies of neuron that is a ganglion. This is also a collection of cell bodies of neurons, so that's another ganglion. The neuron that come preceding the ganglion is therefore preganglionic, and the neuronal process beyond the ganglion is therefore postganglionic. So that's why we give them those names, pre and postganglionic. All preganglionic neurons, in this case the red ones, all preganglionic neurons are cholinergic. What does that mean? It means that all preganglionic neurons release acetylcholine as their neurotransmitter. I'm using the letter ACH here to refer to acetylcholine, even that one. So all of them are cholinergic neurons, which means that the neurotransmitter this neuron releases here at the synaptic clefts here will be acetylcholine. That therefore means that we'll expect to have receptors of acetylcholine on these postsynaptic membranes, or even this one. We need to have the receptors of acetylcholine there, because the neuron that is bringing the impulses is releasing acetylcholine to the synaptic cleft there. Now that begs the question, what is the role of this ganglia? The plural of ganglion is ganglion. From the drawing, you can see that I've intentionally drawn one single neuron up to the ganglion. Then from the ganglion, I have drawn like 3, in this case, postganglionic neurons. That is very intentional to tell us the kind of things that happen at the ganglion. The main thing here is that the ganglia are sites of amplification. You see only a single neuron coming in, then the effect of that is amplified to multiple other things. So it's a site of amplification. So when you use the term cholinergic, it means that the neuron is releasing acetylcholine. How about the postganglionic neurons? Postganglionic neurons, and that applies to the blue ones, are either cholinergic or adrenergic. We've already defined what is cholinergic. Adrenergic means that they release norepinephrine as the principal neurotransmitter. So the postganglionic neurons can either be adrenergic or cholinergic. Now there's something I want to get there. The postganglionic neuron determines a lot the eventual physiological effects that take place within the target organ, because they're the ones that reach the target organ. So the type of chemical they release, therefore, becomes very important physiologically. That will determine how that organ actually responds. Because you've seen that all preganglionic neurons are cholinergic. Therefore, we can say it actually doesn't matter as long as there's a signal. But the type of chemical postganglionic neuron releases is important in determining the physiological effects that are anticipated here. So some of them could be cholinergic, as shown there, and some of them could be adrenergic. Adrenergic means they release norepinephrine as the neurotransmitter. Having given that general organization, now let's narrow down to the organization of the sympathetic division. Neurons with sympathetic function will arise from the T1 to L3 segments of the spinal cord. That is where the preganglionic neurons arise from. Sorry, down here, T1 segment to L3 segments of the spinal cord. For that reason, we say that sympathetic has a thoracolumbar outflow. Let's look at it from this image. So if that is the brain and this is the spinal cord, we have the cervical segment of the cord giving you cervical nerves, thoracic segment of the cord giving you thoracic nerves. It is this thoracic and upper lumbar segments of the spinal cord that give rise to preganglionic sympathetic neurons. So we say that sympathetic has a thoracolumbar outflow. Importantly, the preganglionic sympathetic neurons tend to have short segments, short lengths. Let me put it this way, that the sympathetic neurons, sympathetic division, tend to have short preganglionic neurons. And that would therefore mean that the postganglionic neurons tend to be long. If you look at this is the preganglionic neuron, it's synapses somewhere there. Then the postganglionics go to the effector organ. So sympathetic division have short preganglionic and long postganglionic neurons. Another important thing is this. The preganglionic sympathetic neurons, wherever they synapse, is the ganglion, of course. The ganglia of sympathetic nervous system tend to interconnect with each other. That interconnection with each other is called the sympathetic chain. You can call it the sympathetic trunk. Having mentioned that the preganglionic neurons are short, it means that the ganglia are actually very close to the vertebral column. Indeed, they are on either side of the vertebral column, right and left. And perhaps this image shows that. So that's a ganglion, that's another ganglion, that's another ganglion. They are very close to the vertebral column, which will be somewhere here. We can also appreciate how the ganglia are interconnected to form a chain of ganglia on either side of the vertebral column. The sympathetic ganglia are paravertebral, and they interconnect to form what we call the sympathetic chain or the sympathetic trunk. I've told you that they have short preganglionic neurons and long postganglionic neurons. The postganglionic sympathetic neurons are predominantly adrenergic. There are a few exceptions to that rule, but most of them are predominantly adrenergic. We have defined what adrenergic means, so we understand. I may give you some exceptions as we go along, but for the sake of us understanding pharmacology of the autonomic nervous system, understand that sympathetic system are just generally adrenergic. And this is what will therefore give us the eventual physiological effects that we see in the sympathetic division. So why do we need the sympathetic division? We need the sympathetic division to excite us under emergency situations, because that excitement tends to really activate the body to utilize its energy. We say that sympathetic division is catabolic, which means energy consuming. Think about those emergency situations. The best example I could give you is, imagine wherever you are right now, a cobra has just appeared where you are. How will you respond? You'll most likely run away, and that's a flight response. You may also decide to just attack the cobra, and that's a fight response. So under emergency situations, we either fight back, attack, or run for your life, flight. Therefore, we say that sympathetic gives you the flight or fight response. Some people usually add another one, the fright, which means you are scared. So fight, fright, and flight response. If you come from some places, you might not pick the difference between what I've said. Fighting back is defending yourself. Flight with L is running away. And fright with the R is you are scared. So when you see that cobra, you'll be so scared. You may decide to fight, or you may decide to run away. These are the responses that sympathetic system gives us. As we think through that, I would want you to imagine in that situation or fight, flight, and fright response, how will different organs in your body behave? How will your heart behave, lungs? And look at the behavior as the behavior that help you to either fight that cobra or run away from that cobra. That would be the exact sympathetic effect. So if you behave in such a way that it doesn't help you either for fighting or running away, that is not necessarily a sympathetic response. Like some people may see the cobra and have diarrhea. That diarrhea does not help you in any way in fighting the cobra or running away. So it's not a sympathetic response. It's another thing. Okay, the effects of sympathetic nervous system are mediated by two major chemicals. We have adrenaline, which is a hormone, you can call it epinephrine. This adrenaline is produced by the adrenal medulla. It comes from the medulla of the adrenal gland. The medulla of adrenal gland is usually stimulated concurrently when the sympathetic system is activated. And so in response, when you activate the sympathetic nervous system, there's a hormonal component of it, which is the secretion of adrenaline from the adrenal medulla. This hormone is released into the bloodstream. And so it goes to many parts of the body. Now, apart from that adrenaline, we're also talking about noradrenaline. Noradrenaline is released from the nerve terminals of the postganglionic sympathetic neurons. So these two chemicals are important in mediating the sympathetic responses. Please note, adrenaline is largely a hormone, so it is circulating within the bloodstream. Noradrenaline is largely a neurotransmitter, so it is within nerve terminals and it will be found in synaptic clefts. The receptors of the two are, however, the same, and the physiological effects of the two are also the same, in as much as maybe they have different likings. Something will stimulate another organ stronger than another one, but they both stimulate the same organ anyway. For the adrenaline or noradrenaline to mediate their physiological effects on their target organs, they must activate the receptors on their target organs. The receptors of adrenaline and noradrenaline are collectively termed adrenoceptors. So adrenaline must bind onto the adrenoceptors for it to mediate its physiological effects. Noradrenaline also must bind to its receptors, the adrenoceptors, for it to mediate its physiological effects. That brings me therefore to this one. What are the adrenoceptors? So adrenoceptors are receptors for adrenaline and noradrenaline. You'll be finding them in the postsynaptic cell, basically. There are two types of adrenoceptors. We have the alpha adrenoceptors and the beta adrenoceptors. The alpha adrenoceptors are of two subtypes. We have alpha 1 and alpha 2. The alpha 1 adrenoceptors are mainly found on the walls of most blood vessels. And I'm not saying all, I'm saying most. The alpha 2 adrenoceptors are found in the digestive system. Well, alpha 2 adrenoceptors are also found in the central nervous system, but we'll not look at that central component because we are looking at autonomic nervous system. So largely we are focused on the peripheral process. The beta adrenoceptors are usually three types, but I'll teach you two, the beta 1 and the beta 2, because the beta 3 is not so much in the autonomic nervous system. Beta 1 adrenoceptors are the ones located on the heart wall. Beta 2 adrenoceptors are found in the airways. We also find them in the walls of coronary vessels, and we find them in the walls of the blood vessels within skeletal musculature. This is a good moment for you to ask yourselves why this kind of blood vessels may require a different type of receptor compared to most of the other blood vessels. Think about the effects that you're going to have as a fight or flight response. How these blood vessels are expected to behave in such conditions would be very different from how you expect these two types of blood vessels to behave in such similar conditions of emergency. It is for that reason that we expect them to behave differently, that we actually need to have different receptors for them. We are not going to fill this column right now because I have another column for that. I prefer talking about the physiological effects towards the end. So I'll skip that last column and we'll revisit it later when you look at the physiological effects of both sympathetic and parasympathetic. So that was the general organization of the sympathetic division. Now let's look at the general organization of the parasympathetic division. Parasympathetic nerves come from either the cranial nerves or sacral nerves. I showed you this image earlier. They come from either cranial nerves or sacral nerves. The ones that come from, so just before I go to that, because they come from either cranial or sacral, we say that parasympathetic has a craniosacral outflow. Craniosacral outflow. The cranial nerves which are parasympathetic are the following. We have the third cranial nerve. We call it the oculomotor nerve. That nerve is parasympathetic to the eye. We have the facial nerve, the seventh cranial nerve. The seventh cranial nerve, facial nerve, is parasympathetic to the lacrimal gland, and it is also parasympathetic to some salivary glands. The particular salivary glands that it's parasympathetic to are the sublingual gland and the submandibular gland. Sublingual and submandibular. The ninth cranial nerve is glossopharyngeal nerve. This one is parasympathetic to the parotid gland. The parotid gland is one of the salivary glands. It's the largest, actually. Then the 10th cranial nerve is also parasympathetic. It is parasympathetic to multiple organs: the heart, the respiratory tree, the digestive system, the urinary system, and even some parts of reproductive system. Now, with that, we see that it is parasympathetic to multiple organs. That is why we call it vagus nerve. The nerve is vague. It supplies multiple things. Okay, how about the sacral nerves? The sacral nerves, which are parasympathetic, are specifically the middle three sacral spinal nerves. So we have S2, S3, and S4. These sacral parasympathetic outflow is targeting the pelvic visceral organs. So think about the pelvic visceral organs. That is what is targeted by the parasympathetic outflow from the sacral segment. Right. Importantly, the parasympathetic nerves have very long preganglionic neurons, as we can see here. The preganglionic neurons are very long. Because the preganglionic neurons are long, it means that the ganglia is located very near the organ, or sometimes within the organ wall itself. As you can see in these images, the red represents the preganglionic, which is cholinergic. Then the black ones represents the postganglionic neurons. So some of them, the ganglion is actually within the wall of the organ being innervated, and some of them, the ganglion is near the organ. Either way, we realize, therefore, that the postganglionic parasympathetic neurons are very short. So we can say that the ganglion is so much away from the vertebral column. And the term we use for that is the ganglia are peripheral. Both preganglionic and postganglionic parasympathetic neurons are all cholinergic, which means that parasympathetic division mediates its physiological effect by the release of acetylcholine as a neurotransmitter. So parasympathetic effects are all cholinergic effects. The postganglionic neuron releases acetylcholine. Now that means that acetylcholine will therefore act by stimulating its receptors on the target organs. The receptors of acetylcholine are termed cholineceptors. The functions which are parasympathetic are largely concerned with the vegetative aspects of day-to-day life. You know, the things that maintain life, basic minimum. Just maintain the basic metabolic rate. Things like breathing, relaxing, and things like that. Generally, we see that parasympathetic functions conserve body energy rather than utilize body energy, as we saw in the sympathetic. We generally look at parasympathetic as a rest and digest kind of response. And how you want to look at this is this way. So in sympathetic, we said, imagine that a cobra has just appeared where you are. In sympathetic, I want you to imagine that you've overeaten, and so you're just relaxing. And there's no exam. How do you behave? Just rest and digest it. Those are the kind of responses parasympathetic can give you. So as we'll be looking at organ system effects, I want you to think through those. Think through how do you behave when you've overeaten? How the different organs in the body behave when you've overeaten and you're just resting? That will be parasympathetic. And for sympathetic, how do you behave when you see that cobra? Okay, now I want us to talk about the cholineceptors, the receptors of acetylcholine. But give me a second again. Someone has. Cholineceptors are receptors of acetylcholine. What are the different types of cholineceptors? There are two different types of cholineceptors. We have the nicotinic receptors, and we have what we call muscarinic receptors. The nicotinic receptors are stimulated by nicotine, or at least in the discovery or in the experimental setup, they are noted to be stimulated by nicotine, and that's what they're called nicotinic receptors. There are two types of nicotinic receptors. There's what we call N1 receptors, or others called muscle-type nicotinic receptors. Muscle-type nicotinic receptors are found on skeletal muscles. So these ones are not necessarily autonomic in function because they are found on skeletal muscles. If you remember the story about neuromuscular junction, we talked about acetylcholine being released into the neuromuscular junction and that it binds on receptors on the muscle. Those receptors are muscle-type nicotinic receptors. Therefore, N1 receptors are not necessarily part of autonomic system, but part of the somatic nervous system. But I like mentioning it here just to help you understand that even though we are talking about acetylcholine under sympathetic and parasympathetic nervous system, acetylcholine is not necessarily limited to the autonomic nervous system. Acetylcholine also is in the somatic nervous system. So don't think that acetylcholine is only limited to the autonomic nervous system. That's why there's some drugs that we give that target acetylcholine or its receptors that will also affect the rest of the nervous system, not just the autonomic system. The second type of cholineceptors are called neuronal nicotinic receptors. Neuronal nicotinic receptors, also called the N2 receptors, are found in autonomic ganglia. These are the junctions between the preganglionic and the postganglionic autonomic neurons. So that's where they are found. So these ones are autonomic by far. Then we have the muscarinic type of receptors. There are different types of muscarinic receptors. There's M1, M2, M3, M4, M5. But I don't want us to go in that direction. Let's just generalize and say we have muscarinic receptors. They are found in the wall of the heart, they are found in smooth muscles, and they are found in exocrine glands. What do you notice? The muscarinic receptors are actually the ones that we really want to know because it actually summarizes the target organs of the autonomic nervous system, those three. They all have muscarinic receptors. And so the effects of acetylcholine, the effects of parasympathetic division will be largely mediated by this kind of stimulation if we stimulate the muscarinic receptors. Having talked about the organization of sympathetic division and organization of parasympathetic division, let's now say some things about the anatomical differences between the sympathetic and parasympathetic division of the autonomic nervous system. We can compare a number of things. One is their origin from the central nervous system. We've seen that sympathetic division comes from thoracic and lumbar segments, while parasympathetic arise from cranial and sacral nerves. So sympathetic is thoracolumbar outflow, while parasympathetic is a craniosacral outflow. We can compare the length of the preganglionic neurons. We noted that the sympathetic ones are shorter compared to parasympathetic. We also noted that there's a difference in the length of the postganglionic neurons. The sympathetic ones are longer compared to the parasympathetic ones. We can compare the location of the ganglion. The sympathetic ganglia are near the vertebral column because the preganglionic neurons are short. So the sympathetic ganglia are paravertebral. The parasympathetic ganglia, we mentioned that they are peripheral, away from the vertebral column. And lastly, we can compare the concept of ganglia communication. Sympathetic system, the ganglia communicate to form the sympathetic chain. We don't have such communications in the parasympathetic division. So those are the differences between sympathetic and parasympathetic division of the autonomic nervous system. So when you look at the physiological differences between the parasympathetic and sympathetic divisions of the autonomic nervous system, there are also a number of parameters that we can check. One of the parameters we can check is the type of postganglionic neuron that we have. The sympathetic one is adrenergic, at least for most of the cases. The parasympathetic one is cholinergic for all cases. We've also seen that we can compare the type of responses that we have. Sympathetic gives you flight and fight response, while the parasympathetic gives you rest and digest kind of responses. You can compare the involvement of the endocrine system. Sympathetic involves the endocrine system. The adrenal gland produces adrenaline hormone into the bloodstream when the sympathetic system is activated. We don't have such in the parasympathetic. You can also compare the distribution of the effects. Now, I want you to understand this way, that the sympathetic effects are diffuse. That means that they affect multiple organs at the same time. Now, why? Two reasons why sympathetic effects are diffuse. One is on the amplification that occurs at the ganglion. We saw that we have preganglionic neuron synapsing with postganglionic neurons. The sympathetic division has higher amplification. The preganglionic neuron synapses multiple postganglionic neurons. And so the effects are going to be diffuse. If you stimulate sympathetic system, you affect multiple organs. Other than the amplification that takes place, the other are diffuse is because of the involvement of the endocrine division. Because we involve the endocrine division, because we involve the adrenal gland, it means that adrenaline is being released into the bloodstream, and that goes to multiple organs, so therefore making the sympathetic effects diffuse. On the other hand, the parasympathetic effects are localized. That is to mean that they target specific organs. Last but not least, we compare the usage of body energy, and we mentioned that sympathetic is catabolic, energy consuming, while Sympathetic is catabolic, energy consuming, while parasympathetic is anabolic, energy conserving. Great. So I want us to now look at the organ system effects of both sympathetic and parasympathetic divisions of the autonomic nervous system. And there are a number of organs we are going to look at. I want to start with the eye. Think about the eye. The eye has some smooth muscles within it in the iris. The role of the iris is to control the size of the pupil. Under sympathetic effect, the pupil dilates to allow more light to enter, while under parasympathetic effect, the pupil constricts. Now, you don't have to cram this one. Think through what I told you earlier. How should your pupil behave when you see a cobra? And how should it behave when you just overeaten and there's no exam? So when you see a cobra, that emergency situation, the pupil dilates. When you don't even have to see far, the pupil constricts. Also in the eye, we have the ciliary body which control the shape of the lens. When you see a cobra, you need to see far. For that reason, the lens will flatten as an adaptation for far vision. But when you just overeaten, you don't even have to see far, maybe just up to your TV. And so the lens will converge, and that's an adaptation for near vision. We call that accommodation. We see in the eye that sympathetic and parasympathetic have dual innervation with antagonistic effects. How about in the cardiovascular system? We can start with the speed at which the heart beats. We call that heart rate, the number of beats per minute. Sympathetic will increase the heart rate, and that's what we call positive chronotropic effects, while parasympathetic will decrease the heart rate, and that is what we call the negative chronotropic effect. I want you to understand that increasing heart rate here does not necessarily mean tachycardia. If the heart rate was 60 and it has gone to 80, that is still a positive chronotropic effect. But it is also true that it may be 80 and it has shot to 110. That is still positive chronotropic effect, and in that case also tachycardia. So here we're just looking at the increase in the heart rate. About the contractility of the heart, sympathetic makes the heart to pump with the higher force. The contractility is increased, while parasympathetic makes the heart to not contract with a stronger force. The term given to contractility is inotropy. So sympathetic has a positive inotropic effect, while the parasympathetic has negative inotropic effects. Lastly, on the heart, we look at the conduction velocity. What speed are the impulses traveling with from the sinoatrial node to the rest of the musculature of the heart? The speed of impulse conduction is increased in sympathetic stimulation and decreased in parasympathetic stimulation. The term given to conduction velocity is dromotropy. So sympathetic has a positive dromotropic effect on the heart, while parasympathetic has a negative dromotropic effect on the heart. Other than the heart, we can also look at the blood vessels, and especially the arterioles. For most blood vessels, because of alpha-1 stimulation, most blood vessels will vasoconstrict. Under sympathetic stimulation, but there'll be no effect in parasympathetic stimulation. The reason is because most blood vessels do not enjoy parasympathetic supply. So this is an example of single innervation. While in the heart, examples of dual innervation with antagonistic effects. Now, how about the effect on blood pressure? Because of the effect this one has on the heart and blood vessels, sympathetic raises your blood pressure. So in that emergency situation where you've seen a cobra, your blood pressure may shoot up. And in parasympathetic, the blood pressure will fall, not because of what the parasympathetic has on blood vessels, but what it has on the heart that still contributes to a fall in blood pressure. Right, so this forms part of what we utilize actually from a pharmacological perspective. When you're resuscitating a patient, you give them adrenaline, you're just giving positive inotropic drugs. Or when you want to manage a patient with bradycardia, sometimes you just give them drugs that inhibit the parasympathetic division. And that will elevate their heart rate because you block this one. Okay. I hope you do remember that the sympathetic effects on the heart are mediated by beta-1 adrenoceptors, and that these parasympathetic effects on the heart are mediated by the muscarinic receptors. In the respiratory system, we can look at the diameter of the airways. If you see a lion, that's the time that the airways need to open up. So we have bronchodilation. And under parasympathetic stimulation, there is bronchoconstriction. Now, this forms why we then use drugs which promote sympathetic system in the management of a patient with asthmatic attack. They are bronchodilators. But you can also give a drug that inhibits the parasympathetic nervous system, and that will still cause bronchodilation. If you inhibit parasympathetic system to the respiratory system, or you stimulate sympathetic system to the respiratory system, they both cause bronchodilation. I hope you do remember, however, that the sympathetic effects on the airways are mediated by beta-2 adrenoceptors. Secretions in the airways also change under the two systems. So sympathetic reduces the secretions and parasympathetic increases the secretions. It explains to you why, if you're going to induce general anesthesia on a patient, sometimes we give them atropine to block the parasympathetic system so that we reduce the airway secretions. Because if we are going to give them general anesthesia, if you have secretions there, you may have aspiration. And that's why we block the parasympathetic division to reduce the secretions. Remember that the effects on the airways under sympathetic are under beta-2 stimulation. In the digestive system, okay, before we go to digestive system, also we've noted that in the respiratory system, this is an example of dual innervation with antagonistic effects. How about in the salivary secretion? Very unique. Sympathetic makes you to produce thick saliva, not so much. Parasympathetic makes you to produce watery saliva and a lot of it. How do we call that? We don't prefer calling this as dual innervation with antagonistic effects. We consider those as very different effects. So we call this dual innervation with different effects. That's a good example for dual innervation with different effects. On peristalsis, sympathetic system reduces peristaltic activity while parasympathetic increases the peristaltic activity in the GIT. On sphincter, sympathetic system increases the sphincter tone, which means that the sphincters become tighter. They don't allow things to pass through. While in parasympathetic, the sphincter tone reduces, things can pass through. How about on acid secretion? Sympathetic reduces gastric acid secretion, while parasympathetic increases gastric acid secretion. So on peristalsis, sphincters, and gastric acid secretion, dual innervation with antagonistic effects. Think through this. If sympathetic increases the sphincter tone, which means that the sphincters become tight, and reduce peristalsis, which means that food does not move as fast. It means that you're going to retain food in the GIT for long, so that the stool type will actually be very hard. But in the parasympathetic division, because things are moving faster and the sphincters are relatively relaxed, stool will be soft. Okay, let's look at the urinary system. In the urinary system, renin secretion from the kidney is stimulated by the sympathetic system and reduced by the parasympathetic system. Dual innervation, antagonistic effects. In the urinary bladder, the sympathetic system reduces bladder contraction, which means it causes bladder relaxation, bladder wall relaxation. Parasympathetic increases contraction of the urinary bladder. That's why in patients with the atonic bladder, you can give them drugs that promote parasympathetic stimulation to cause bladder contraction. How about the sphincters for the bladder? The sphincter tone for the bladder increases under sympathetic stimulation, which means that doesn't allow urine to pass. While for parasympathetic, the sphincter tone reduces, the sphincter is now not so tight, urine can pass. Therefore, sympathetic will reduce voiding. That's why when you're running around the field, you are unlikely to feel that you're pressed, especially in a state of emergency. You are unlikely to feel the urge to go for a short call. But when you're in a relaxed state, you can actually feel it. Parasympathetic promotes voiding. In the genitalia, very interesting things we want to see, but in the urinary system, we've seen dual innervation with antagonistic effects. In the genitalia, the blood vessels of the penis respond by vasoconstriction under sympathetic stimulation. That one is not unusual. But the blood vessels respond by vasodilation under parasympathetic system. That's unusual because remember we said that we don't really have parasympathetic nerve supply to blood vessels, but now these are some of the few blood vessels that receive parasympathetic supply, the erectile tissues. So they could be the one for the clitoris or the one for the penis. The erectile tissues have blood vessels which vasodilate under parasympathetic stimulation. If the blood vessels vasodilate under parasympathetic stimulation, it therefore means that will cause erection. And so parasympathetic is the one that causes the penis or the clitoris to erect because of increased blood flow. It should make sense. You've just eaten. That's the time to think about such things. You are just relaxing. As opposed to you've seen a cobra, there's no time to have sex. But very interestingly, sympathetic stimulation to the penis actually causes ejaculation. What do we see here? We see a collaboration between the sympathetic system and the parasympathetic system to help achieve the male sexual response. For that reason, this is an example of dual innervation with synergistic effects. Parasympathetic causes erection, and then sympathetic causes ejaculation or orgasm, so to speak. How about on the gravid uterus? The gravid uterus will be a bit interesting. It usually has adrenoceptors whose activation causes relaxation of the myometrium. It is for that reason that we can actually use salbutamol, for example, to knock off labor process because it causes relaxation. We don't have much of effects of parasympathetic on the gravid uterus. It's an example of single innervation. There are some special occasions that I also want to highlight. One of them is the sweat glands. Sweat glands respond by producing sweat under sympathetic stimulation. This response is unique because it is not adrenergic response, but a cholinergic response. I told you that most postganglionic sympathetics are adrenergic. That is true. And the keyword there is most, which means there are a few exceptions. And innervation to sweat glands is one of those few exceptions where the innervation is sympathetic, yes, but cholinergic instead of adrenergic, and it promotes sweating. We don't have parasympathetic nerve supply to sweat glands. Liver also respond by breaking down more glycogen to release glucose. Remember, sympathetic is catabolic. Parasympathetic may not have effect on glycogenolysis. Adipose tissue also respond under sympathetic stimulation to break down fat, basically to release fatty acid that can be metabolized. Remember, it's catabolic. We don't have parasympathetic effect on the adipose tissue. Lastly, the adrenal gland respond under sympathetic stimulation by releasing adrenaline hormone. Again, the stimulation to the adrenal gland by sympathetic division is not adrenergic, but it's cholinergic. But in response, the gland produces adrenaline into the bloodstream, and that will go to different organs in the body. Therefore, sympathetic effects become diffuse. We don't have parasympathetic supply to the adrenal gland. Good. We've talked about sympathetic division and the parasympathetic division. I told you that autonomic nervous system has another division which we like giving it its special attention. I'll just mention it here briefly, but much of it is handled under physiology of the digestive system. The enteric nervous system is a functionally independent nerve plexus. I want you to add the term plexus functionally independent nerve plexus within the wall of the gastrointestinal tract, or what we call the gut. These nerve plexus control the different functions of the digestive system. It consists of two types of plexuses. There's what we call the myenteric nerve plexus. These are nerve plexus within the muscular layer of the alimentary canal. The myenteric plexus control contractions. So like peristaltic activity or even the sphincter tone is controlled by the myenteric plexus. There's another plexus though, just beneath the epithelium within the submucosal layer. So we call them submucosal nerve plexus. The submucosal nerve plexus control the different type of secretions within the gastrointestinal tract. Could be exocrine secretions most of the time, or sometimes even endocrine secretions. So it's a rich nerve plexus. These plexuses are interconnected within one another and even with the others. They are rich, they are interconnected. They control the functions of the digestive system. Even if they control the functions of the digestive system, the rate of the operation is regulated by the sympathetic division and parasympathetic division of the autonomic nervous system. So where sympathetic and parasympathetic come in is to control the rate of function. Otherwise, even if sympathetic and parasympathetic are not there, these nerves will still operate to control the functions of the digestive system. And that's why we're saying it's functionally independent. It can operate without the central nervous system. It is for that reason that some people call it the enteric brain. So they are interconnected, as you can see in this image, whether submucosal or myenteric, and they receive control or regulation from both sympathetic and parasympathetic division. And that's why we're saying that sympathetic can reduce acid secretion, parasympathetic promote acid secretion. All right, so that is it for today. That is the story of the autonomic nervous system.